Hi, welcome to Sev Science, a channel where you can learn your basic biology concepts to pass your class. In today's lesson, we're going to be talking about Chapter 12.6, Structure of the Eukaryotic Chromosome. Let's get right into it. The eukaryotic chromosome contains a single double helix DNA molecule, but it is composed of more than 50% protein. One protein, called histones, play primarily a structural role. The five primary types of histone molecules are designated H1, H2A, H2B, H3, and H4. The amino acid sequences of H3 and H4 vary little between organisms. For example, the H4 of peas is only 2 amino acids different from the H4 of cattle. This similarity suggests that a few mutations in the histone proteins have occurred during the course of evolution, and that histones have essential functions for survival. A human cell contains at least 2 meters in DNA, yet all of this DNA is packed into a nucleus that is about 5 nanometers in diameter. The histones are responsible for packaging the DNA so that it can fit into such small spaces. First, the DNA double helix is wound at intervals at around a core of 8 histone molecules. There are 2 copies of H2A, 2 copies of H2B, 2 copies of H3, and 2 copies of H4, which all total up to 8 histones. This gives the appearance of a string of beads. Each bead is called a nucleosome, and the nucleosomes are said to be joined by linker DNA. Next, the string is compacted by folding into a zigzag structure, further shortening the DNA strand. Histone H1 appears to mediate this coiling process. The fibers then loop back and forth into radical loops. This loosely coiled euchromatin represents the active chromatin containing genes that are being transcribed. The DNA of euchromatin may be accessed by RNA polymerase and other factors that are needed to promote transcription. Under a microscope, one often observes dark-stained fibers within the nucleus of the cell. These areas within the nucleus represent a more highly compacted form of the chromosome called heterochromatin. Most chromosomes exhibit both levels of compaction in a living cell, depending on which portions of the chromosome are being used more frequently. Heterochromatin is considered inactive chromatin because the genes contained on it are infrequently transcribed, if at all. Prior to cell division, a protein scaffold helps to further condense the chromosome into a form that has characteristics of metaphase chromosomes. The more compact the chromosomes are, the easier it is to move about than an extended chromatin. All in all, the steps that DNA takes to form chromatin is 1. The wrapping of DNA around histone proteins, 2. Formation of a 3D zigzag structure via histone 1 and other DNA binding proteins, 3. Loose coiling into radical loops, 4. Tight compaction of radical loops to form heterochromatin, and then number 5, metaphase chromosome forms with the help of protein scaffold. Thanks for watching, and I hope you learned something new about the structure of the eukaryotic chromosome. Have a nice day.